Hi everyone, my name is Nara and today I will be presenting on epigenetics, exercise, and metabolic disorders. So the purpose of my presentation is to just give a really brief overview about the epigenetic effects of exercise and how these changes can relate to metabolic disorders. So first we will talk about epigenetics and what it is, how it works, and why it's important. And then we will briefly touch on metabolic disorders with special attention to obesity, insulin resistance, and type 2 diabetes, just the characteristics of those three disorders, since those are the three that will be emphasized in this presentation. And then finally, we will discuss the epigenetic effects of exercise on those three metabolic disorders. We'll look at the current research and then talk about the application for exercise and epigenetics for these disorders as well. So to start us off, epigenetics. What is it? It can seem like a scary term, especially if you're not familiar with the field of genetics. So simply, it is the stable, inheritable changes in gene expression that are independent of the DNA sequence. So to help us better understand what epigenetics is, we are going to watch this very brief video that does a really great job of simplifying the concept of epigenetics and making it a little bit easier to understand.
The sweet smell of fruit doesn't normally send rats running, but when researchers paired the orange, cherry, almondy scent of the chemical acetophenone with a painful electric shock, lab rats quickly learned to fear it. Along the way, extra neurons sprouted in their noses and in the smell processing center of their brains, making them super sensitive to the scent. This result isn't shocking. What is surprising is that the rats' pups and their pups' pups were also startled by the smell of acetophenone and had the same extra neurons as their fathers, despite never having been introduced to either their dads or the fruity scent before. But how could the pups have inherited something that their fathers learned? Basic genetics tells us that only DNA gets passed along to offspring. Characteristics like memories, scars, or giant muscles can't get passed on since acquiring them doesn't alter the genetic code. But it turns out that instilling fear in the rats did trigger genetic changes, not in the DNA sequence itself, but instead in how that code was read and used in the rats' bodies. In every cell, biological machinery constantly translates DNA into the proteins needed to carry out vital processes. Chemical switches attached to the DNA turn genes on or off or up and down, telling the machinery which proteins to produce and in what quantities. These switches, called epigenetic tags, are why a kidney cell looks and acts differently than a skin or nerve cell, even though all three cells have identical DNA. But the switches in any one cell aren't set in stone. Teaching those rats to fear the fruity smell switched one of their smell-sensing genes into overdrive. Researchers don't know all the places in the rats' bodies where this switch got flipped, but they know it happened in one key set of cells, the rats' sperm cells, which would one day pass along the tweaked genetic material, making the next generation of rats super sensitive to acetophenone. Rodents aren't the only creatures demonstrating this weird type of inheritance. In Epigenetics, Sweden, boys who suffered through tough winter famines went on to have super healthy sons, with extremely low rates of heart disease and diabetes. And their sons had the same excellent health, living an unbelievable 32 years longer on average than the grandsons of boys who hadn't gone hungry. To be clear, this does not mean that we should start starving our kids for the benefit of future generations. Scientists don't even know yet exactly which switches the Swedish famines flipped. While we have been able to connect specific epigenetic changes to health effects in mice, we're a long way off from being able to make those connections in humans. That may sound like a bummer, but it's mostly because we humans don't live in the well-controlled environment of a laboratory. And for that, we should be grateful.
Okay, so hopefully that video gave us a better understanding of what epigenetics is. Next, we'll discuss how epigenetic changes come about. So, there are three main epigenetic modification mechanisms. The first is DNA methylation, the second is histone modification, and the third is RNA expression changes. First, we will talk about DNA methylation. It plays an important role in cell differentiation, embryonic development, and mediating gene regulation. So in human DNA, we have these things called CpG sites, which are the most common sites of DNA methylation. And they are sites along our DNA sequence where a cytosine or a C nucleotide is next to a guanine or a G nucleotide, hence the C and the G. There are about 28 million CPG sites in the human genome, and the majority of these are methylated. So just because something is methylated does not necessarily mean it's a bad thing. However, we have these things called CPG islands, which are really long stretches of the DNA sequence that have a high content of these CPG sites. So things like this picture shows here. These are typically found in the promoter regions of genes, so things that tell a gene to be expressed or not. And these CPG islands are typically unmethylated. So we can go ahead and look at this image here, and we can see that the top one, it can be considered the normal state, and the CPG island is unmethylated. So gene expression occurs as normal, nothing changes. However, once we methylate that CPG island, we see that gene expression is repressed. So it's like a light switch, it turns the gene off instead of on. Basically what happens in DNA methylation is a methyl group attaches to the cytosine of the CPG site, and that cytosine is then converted to a thymine, or the T nucleotide base, and this can result in gene silencing, so turning the switch off, repressing gene expression. The next epigenetic modification we will talk about is histone modification, and this can occur through multiple different ways, and they are modifications of the histone tail. So these can include acetylation, methylation, phosphorylation, ubiquitination, and simylation. We will pay special attention to histone acetylation in this case because it is the only one really associated with epigenetic changes related to exercise. And histone modifications have a role in altering the chromatin structure as well as changing gene expression. The final epigenetic modification that we will talk about is RNA expression. So in our genome, we have non-coding RNA, which is basically RNA that is transcribed from DNA but is not translated into protein. So it may seem like it's unimportant because it doesn't actually do anything in terms of coding for protein. However, it can affect other things that code for protein. So it can cause a change down the line. If there's a change in the non-coding area, it can affect the coding area. Non-coding RNA is involved in histone modifications, DNA methylation targeting, heterochromatin formation, and can also affect gene silencing. So why is epigenetics so important? Obviously, it shows how our environment and our lifestyle can have an effect on our genes and therefore result in changes to our overall health. And because epigenetics are heritable, meaning they can be passed from generation to generation, they can affect future generations. Next, we will address metabolic disorders, which are normal metabolic processes that are disrupted due to abnormal chemical reactions in the body. And these can result from improper organ function, or they can also have an underlying genetic basis. So the three we will talk about today are obesity, insulin resistance, and type 2 diabetes. So first, addressing obesity. According to the latest CDC statistics, 1/3 of US adults and 17% of children are obese. Obesity has been shown to be related to heart disease, stroke, type 2 diabetes, as well as cancer. Insulin resistance is when the body is able to produce insulin but is unable to use it effectively, which can cause glucose to accumulate in the blood and can lead to type 2 diabetes or cause an individual to become pre-diabetic. Insulin resistance is associated with obesity as well as physical inactivity. And type 2 diabetes is the final metabolic disorder that we will address in this presentation. 9.3% of the American population has diabetes, and there's also a larger portion of the population that has diabetes, but they are just undiagnosed. Type 2 diabetes is the most common type of diabetes, and it accounts for 90 to 95% of all diagnoses in adults. Insulin resistance is a precursor to type 2 diabetes, and obesity and inactivity are once again risk factors for developing this disease. With all three of these metabolic disorders that I just mentioned, a healthy lifestyle, diet, and exercise are the typical ways to reduce risk factors for developing these disorders. And while all of us might be aware of the physical benefits of exercise, we might not be as aware of how exercise can benefit us at the genetic level. How does exercise affect our genes? The first study we'll look at is a very extensive genome-wide study that looked at the DNA methylation patterns in adipose tissue before and after a 6-month exercise intervention. So, the study included 23 healthy men with an average of 37 years old and an average BMI of 27.8, and all of the men had a previously low level of physical activity. Some of these individuals had a family history of type 2 diabetes and others did not. It was pretty evenly split. However, it was determined at a baseline test that family history did not affect any of the factors that they were looking at in this study, so they kind of disregarded the two separate groups and counted it as one. The exercise intervention consisted of an hour of spinning and 2 one-hour aerobic exercise sessions weekly for a total of 6 months. Following this 6-month exercise intervention, all participants experienced a significant decrease in hip-to-waist ratio as well as a significant decrease in waist circumference, both of which are attributed as risk factors for type 2 diabetes and cardiovascular disease. They also experienced a significant decrease in blood pressure and resting heart rate, as well as an increased HDL and VO2 max. So an overall increase in cardiovascular health and reduction of these risk factors that are associated with type 2 diabetes. And on top of all of these physical and cardiovascular changes, participants also had changes to their epigenetic profiles. Researchers looked at nearly 500,000 CpG sites within the adipose tissue and found significant differentiation post-exercise at almost 18,000 CpG sites. And of these sites, they corresponded to about 7,600 individual genes. So that's a lot of genes affected just from 6 months of exercise. Of these affected genes, they found that 18 genes were candidate genes for obesity and 21 genes were candidate genes for type 2 diabetes, meaning that these genes had been previously associated with these disorders. So researchers wanted to investigate this overlap between genes changed by exercise and associated with these disorders even more, so they conducted an in vivo experiment with 2 of the genes that were found to have differentiated DNA methylation post-exercise intervention, as well as being candidate genes for obesity and type 2 diabetes. So, looking at their in vivo experiment, they silenced the genes HDAC4 and NCOR2 in adipocytes. So these genes had an increased DNA methylation level and a decreased mRNA expression level after exercise, as measured during the pre- and post-exercise intervention portion of the study. And then in the cultured adipocytes, researchers found that silencing these genes resulted in reduced HDAC4 protein levels and reduced NCOR2 mRNA levels, so reductions in both of these gene expressions. This reduction also led to an increase in lipogenesis. So, HDAC4 and NCOR2, what are they? What do they do? Why do we care about them? So they are both biologically interesting candidate genes for obesity and type 2 diabetes. And they both play roles in adipose tissue metabolism. Talking about HDAC4 first, which is a histone deacetylase enzyme, so it's involved in that histone modification that we talked about earlier as an epigenetic modification mechanism. Research done by Weems, Griesel, and Olson in 2012 found that this particular gene repressed GLUT4 transcription in adipocytes. And GLUT4 is an insulin-regulated glucose transporter found primarily in adipose tissue and striated muscle. Additionally, McCoy, Proieto, and Hargreaves found that GLUT4 typically decreases in response to exercise, and defects of the GLUT4 gene are associated with insulin resistance. So it's very closely associated with these metabolic disorders that we're focusing on. The increase in lipogenesis after silencing HDAC4 that Rohn et al. Found as part of this in vivo experiment could indicate a reduced repression of GLUT4, which would lead to an increase in glucose uptake in adipocytes and the incorporation of glucose in triglycerides during the process of lipogenesis. Next, talking about that other gene involved in this in vivo experiment, which is NCOR2. It is a nuclear co-repressor, and it is typically involved in the regulation of genes related to adipogenesis as well as lipid metabolism. And a study by Watson, Farrell, and Schwabe found that it has the ability to recruit those histone deacetylase enzymes, including HDAC4, which is the other gene studied. And research done by Galmosi et al. Found that HDAC inhibitors could be potentially used as treatments for obesity and type 2 diabetes. The next study we'll look at investigated insulin resistance in obese individuals and how epigenetic regulation of mitochondrial DNA was involved. So, mitochondria are the foundation for metabolism in human cells, and they can also undergo epigenetic changes because mitochondria have their own DNA. This study looked at mitochondrial DNA, or mtDNA, and the mitochondrial DNA copy number, or mtDNA-N. And they had 40 participants, 32 of which had a BMI greater than 30 and were considered the obese category, and 8 of them had a BMI less than 25, and these individuals were considered the lean category. So what researchers found was that obese individuals had impaired insulin signaling and that they also had a decreased mitochondrial DNA copy number compared to their lean counterparts. So if we look at the figure here, which is the mitochondrial DNA copy number, we can look at the comparison between the lean group and the obese group, and it was adjusted for the difference in group numbers. And we can see that there was a very significant difference between the two groups. Previous studies have also shown that decreased mitochondrial DNA copy number, in addition to obesity, has also been linked to type 2 diabetes, and this deficit can occur in muscle tissue, adipose tissue, as well as in the blood. So, they also looked at insulin-resistant individuals compared to insulin-sensitive individuals in this same study, and they found that those who were insulin-resistant also had lower mitochondrial DNA copy numbers compared to those who were insulin-sensitive. So once again, we have a nice figure here. It was less significantly different than before, but still significantly different. In addition to finding differences in mitochondrial DNA copy number between obese individuals and those who are insulin resistant, researchers also looked at DNA methylation patterns within the mitochondrial DNA. Specifically, they looked at the displacement loop, or the D-loop region of the mitochondrial genome, which is important for controlling the replication and transcription of mitochondrial DNA. What they found was that individuals who were obese or insulin resistant had higher levels of DNA methylation within the mitochondrial D-loop compared to those who were lean or insulin sensitive. So, decreased mitochondrial DNA copy number could be attributed to this increase in DNA methylation in mitochondria, and these results are ultimately important because it helps us better understand how mitochondrial epigenetics, in addition to regular epigenetics, could affect metabolic regulation. So that study paved a connection between mitochondrial epigenetics and metabolic disorders. However, it didn't really address the exercise side of things. So how does exercise affect mitochondria and mitochondrial epigenetics? A 2006 study done by Menchikova et al. Did an exercise intervention with elderly individuals. It consisted of 8 elderly subjects with an average age of 67 years old. 3 females and 5 males, none of which were currently engaged in regular exercise, had no cardiovascular disorders, no diabetes, and maintained a stable weight. The exercise intervention consisted of 12 weeks of training containing 4 to 6 sessions a week. It was moderate intensity, and it was a progressive program broken into 3 phases, so 3 4-week phases, and each program was prescribed individually for each subject. At the end of the exercise intervention, researchers found significant improvements in insulin sensitivity as well as increased mitochondrial DNA, indicating an overall increase in mitochondrial number within the skeletal muscle. So, if we look at this figure here, we can see that this is the pre-exercise and this is the post-exercise for the mitochondrial DNA copy number, and we can see that the post-exercise measure is significantly higher than the pre-measure. Additionally, researchers looked at mitochondrial electric transport chain activity measured by NADH oxidase levels, and they found a significant increase in NADH oxidase, which indicated an overall improvement in mitochondrial function. So, once again, we see a significant difference between the pre- and the post-exercise intervention measures. So, this study showed how exercise can increase the mitochondrial DNA found within skeletal muscle, thus indicating an overall increase in mitochondrial volume. And it also showed how exercise can improve the function of mitochondria in elderly individuals. Additionally, it showed how exercise can improve insulin sensitivity, which is what we talked about in the previous article. It really shows how exercise can be a preventive as well as therapeutic measure for these metabolic disorders that we're addressing in this presentation, not just at a physical or physiological level, but even at a genetic level. Recall from that video that we watched at the beginning of the presentation that epigenetics can be inherited, so they can be passed from generation to generation. So related to that concept is this next study that we'll talk about, which showed how a mother's diet during gestation can affect epigenetic changes in their offspring. This study looked at the gene HNF4A, which is crucial to fetal pancreas development as well as adult pancreas function and therefore glucose tolerance. So in both rats and humans, it is transcribed primarily by the P2 promoter. So researchers in this study took mother rats and fed them either the control diet consisting of 20% protein or the low protein diet consisting of only 8% protein. The mommy rats ate these special diets during pregnancy and lactation, and once the pups were weaned, the pups were returned to a normal rat diet. So, the diet protocol had been used before and had previously been shown to program type 2 diabetes in the low protein group of pups during some point of adulthood, usually occurring at about 17 months of age. Researchers found that the pups born to the mothers who were fed a low protein diet had significantly reduced HNF4A levels in the pancreatic islets compared to the control group. Additionally, they found that the islets had significant increases in DNA methylation in the P2 promoter and extensive histone modifications for the low protein pups. Thus, the maternal diet caused epigenetic changes in the pups, which eventually caused them to develop type 2 diabetes later in their life due to that dysfunctional gene because of the low protein diet. In conclusion, epigenetics is a link for us between our environmental and lifestyle choice interactions and how they affect our genome. So you might have heard of gene-environment interaction. Epigenetics is kind of the result of that. Additionally, these studies have showed us that exercise is not just beneficial for our physical selves, but can also be used as a preventive tool and a therapeutic tool for these metabolic disorders. I mentioned briefly in one of the studies that epigenetic modifications are being investigated further to see if we can develop therapies for such disorders and diseases. So DNA methylation markers can be used to target specific genes affected by epigenetics that cause disorders and diseases in individuals, as well as targeting different types of histone modifications. Since epigenetic changes are heritable, changes that occur in you can affect your future generations. So it's important to note that adapting a healthy lifestyle, exercising, eating a balanced diet, all that kind of thing, they don't just affect you now, but they can eventually affect your future generations if they end up causing major epigenetic changes that are passed on to your offspring. So hopefully this presentation helped you recognize how important exercise is, even at a genetic level, and how adapting a healthy lifestyle today can not only affect you now or down the road, but can also affect future generations. Here are my references, and thank you for watching.